Kind word. It's my honor actually to be here and to all the chair at the University of East Anglia. And thank you all for coming and also for watching. So as the title of my presentation mentioned, I am a plant immunologist. And these are two words that maybe you are not used to see together. And I hope that by the end of my presentation, you will understand why it is fair to say that indeed I'm a plant immunologist. The other thing that you will realize, hopefully, is that I'm very keen to understand signaling processes in plant in particular, but in general in any organism, and also to see how we can apply this knowledge for biotechnological purposes, as mentioned by David before. So the first question you may wonder is, why is someone working on plant? And is it still a rare species in science? It's also only a small percentage of the biologists around the world. But actually, it's important to remember that without plants, none of us will be here today. And plants are key for humanity. They provide oxygen, which is required for our survival. They provide food and drinks. They provide material. Actually, probably none of this building would be here without plant, directly or indirectly. They provide fuels, again, mostly indirectly. And they also provide high-end products coming from their secondary metabolism, which often provide drugs, at least in the first instance, before they are synthesized, or also cosmetics. So, as David mentioned as well, we are now facing a great challenge in the 21st century, which is how to achieve food security. How are we going to feed this increasing world population, which has tripled over the last century? And one major constraint to food production around the world, to agriculture, is actually posed by plant pathogen. And this is the topic of the Institute of the Sensory Laboratory where we are working to try to understand indeed how these pathogens are causing disease, how they manipulate the plant immune system, and how we can provide solutions to actually fight this disease affecting important crops worldwide. Here I just want to provide one example, which whether we agree or not with intense agriculture, it's something that we have to keep in mind. This is actually a field of soybean in Brazil, and if you will see the scale, this is actually kilometers of monocultures, of soybean monocultures in Brazil. And in order to maintain this production, and which is thanks to the climate and also the fertility of the soil, enable actually several cropping of soybean in one year. What you can see here is actually tractors collecting the bean from the soil, and here immediately behind, tractors that are immediately replanting the seed for the next generation, the next culture. And in order to enable such intensive agriculture in this condition, this requires very regular and massive usage of chemicals, many of them actually being here to control one single disease, which is called from a fungal pathogen. And this disease is causing this pathogen is causing this disease, which is called Asian soybean rust. So this illustrates really the importance of plant pathogen and the direct impact, economic and environmental impact, that these diseases can have in the world. But you don't need to go as far as Brazil to remember the importance of plant diseases. And this is an example which is in UK and actually very local, even in Norfolk here, where ash dieback, or actually our ash trees, at the moment dying all around the country due to a fungus which has come in recent years from the continent and which is currently devastating English woodlands. So a few years ago, actually, Sarah Gurr and Exeter and colleagues have looked at all the important crop diseases around the world and have tried to bring a cost in terms of human lives of how these diseases are affecting the world population. And they put all these numbers together and they came up with these numbers that if we were able to control only a few of these major diseases, we could actually be able just by itself to feed several hundred millions of human beings. And so this is just to illustrate the often neglecting importance of plant diseases on agriculture and also the neglected importance of agriculture, and where a lot of funding is going to solving cancer or other diseases of humans. It is also important that without food, none of us will be here, and so it's equally important to solve problems that affect the production of food. So, as a community of scientists, it is clear that we need to understand the plant immune system, because without this understanding, it is very difficult to understand how plant pathogens are in the first place causing disease. And also without this knowledge, it is difficult to come up with solutions to actually fight with different diseases. And so here comes the importance of studying the plant immune system. But before going into the specific of the plant immune system, I wanted to go back here to this textbooks example, and I just took this schematic from the Nobel Prize webpage. And this just illustrates the dichotomy of immunity as we have been taught immunity at school, where we have the innate immune system and then we have the adaptive immune system. And I think we all remember that we have this dichotomy in the immune system. But this is an extremely human-centric point of view of the immune system. It's actually specific to mammals. Most higher eukaryotes only rely on the innate immune system. And it is for the discovery of the receptors which are involved in innate immunity in mammals, in mice, and humans, that in 2011, the Nobel Prize for Medicine was actually awarded to two scientists, Bruce Beutler and Jules Hoffman, who discovered these receptors. These receptors here are the plasma membrane, and they are called toll-like receptor. And this receptor are nothing else than a mechanism to perceive non-self, to perceive something which is potentially dangerous, which just signals the presence of a potential pathogen. And as I mentioned, this type of immune receptor are present in all higher eukaryotes, even in the absence of an adaptive immune system, which is unique to mammals. So coming back to plants, indeed, they only rely on innate immunity for their defense. And all cells of plants are able, in theory, to perceive a potential danger. So it's a cell-autonomous immune system. There is no antibodies, again, a major difference. But yet the plant is able to recognize a potential pathogen both locally, so directly at the point of contact between the pathogen and the plant, but also in response to this local perception to induce systemic responses in other parts of the plant which hasn't seen this pathogen, and to prime the response of the plant to a further attack by a pathogen. There is also evidence in the literature, although it is somehow controversial, of a potential transgenerational memory of the stress, where one plant would have responded to an attack, and then the progeny of this plant would be more ready, if you want, to respond to a new attack. So if I try to put that into a simple schematic, which represents our current understanding of the major concept underlying plant immunity, this is represented here. And the only thing I want you to remember from this slide, I will not go into the specific, it's simply that plants rely on two types of immune receptor. The first one is present here at the surface of each cell at the plasma membrane. The second class of immune receptor is present within the cell. The function of these two classes of immune receptor is exactly the same, is to perceive non-self, is to perceive danger. This immune receptor at the surface of the plant are typically referred as pattern recognition receptor. This is a term that we borrow from our colleague immunologists in the animal field. And they perceive conserved molecules from microbes, which are called PAMs, or pathogen-associated molecular pattern. But these are nothing else than non-self determinants. In order to be pathogenic on a given plant, successful pathogens need either to evade this recognition at the surface or to actively suppress it. If they cannot do that, they will not be able to infect a given plant. And this is the best illustration of the importance of this first layer of immune recognition. So in order to become pathogenic, I mentioned that one way successful pathogens are using is actually to suppress actively this first layer of immune recognition. Many pathogens do that by secreting proteins or small molecules that are called effectors. And one of the functions of these effectors is actually to directly suppress the receptors here, potentially by degrading them, as it has been shown in a few examples, or actually by suppressing downstream signaling. But by doing this and basically enabling pathogenicity, they then make themselves susceptible to the second layer of immune recognition in plant, which is mediated by this intracellular immune receptor. And many of them are actually similar to the type of immune receptor that we humans have in our innate immune system, which are called NOD-like protein, and therefore we call them NLRs. So these are the two classes of immune receptor that plants employ to recognize non-self. And in my laboratory, we specifically are studying how this immune perception at the plasma membrane is working at the molecular level, which type of receptors are being used, which type of signaling events, once they perceive a potential danger, are triggered downstream, and how ultimately this translates into blocking the infection by potentially pathogenic microorganisms. So I just want to spend a few minutes just to make parallels between what we know in innate immunity in plants and what we know in animals. So as I mentioned, the Nobel Prize in 2011 was given for the discovery of this class of immune receptor at the plasma membrane. In mammals, they are called TLRs, for toll-like receptors, and they are called toll-like receptor because they are similar to an initial receptor which was identified in insect, which is called toll. In response to perceiving their corresponding ligand, which are either microbial-derived ligand or host-derived ligand, which are released upon infection, for example, these receptors are recruiting a number of adapter proteins within the cell to ultimately lead to the activation of kinases, which are involved in the transmission of the immune signal. In plants, these adapters actually don't exist as such, and these immune receptors are not fused to any of this adapter domain. Instead, plant immune receptors are directly fused to a kinase domain, which phylogenetically, it's actually related to the kinases here involved downstream with innate immune signaling in both mammal and insect. So in a way, it seems that while the general mechanism and concept are conserved between animal and, in this case, plants have managed to sum up, if you want, all of this event within a single protein. And so a lot of my colleagues, or you can read a lot of books or reviews about that, who make the claim that there are a lot of similarities between the immune system of plant and mammal. I actually disagree with that. And if you look at this schematic where it has been attempted to summarize this knowledge, you can see that indeed globally the pathways of the concept are the same. Indeed, there are immune receptors at the plasma membrane, there are a number of signaling events, and ultimately you will have immunity. Some domains are sort of conserved, so both organisms will use kinases, for example. Some of them will use also a tier domain as an adapter. However, the way these domains have been combined in this pathway are different. And all of that actually argue for convergent evolution. It just makes sense to use similar domain for recognition because they are just excellent domain to fulfill this function biochemically. A leucine-rich repeat domain, which is often present in this immune receptor, is, for example, extremely good at binding protein and peptide. So it makes sense to use it to recognize a non-self-determinant, which is indeed a protein. A kinase, it's a very good way to transmit a signal from one protein to another. And so this is why it has been recruited independently. And there are many, many other examples as to why this similarity is actually caused by convergent evolution rather than sharing a common ancestor where an ancestor, if you want, immune kinase, has been conserved and then evolved independently in this lineage. So coming back to plant, for more than 2 decades, physiologists and biochemists have actually identified a number of molecules that, when applied onto a plant cell, will induce immune responses. They're coming from all different classes of molecules. They could be lipid, protein, or carbohydrates. And here, I've just represented these different molecules depending from which organism they were initially identified. We know many non-self-determinant for oomycetes, which represent a major class of plant pathogen, as well as from fungi, bacteria. Recently as well, different molecules from viruses have been shown to be recognized by this surface localized immune receptor in plant. But importantly, it's also important here to note that plant can also recognize molecules which are coming from the plant itself. And these are typical danger signals, which are molecules which normally are not available for recognition by the plant and only become available for recognition upon damage, such as wounding, or in response to infection, where they will be actively produced and secreted in the extracellular space. Also in recent years, pests, so animals, have been shown actually to be recognized by this surface localized immune receptor. And we now know molecules from insect of a nematode that will activate such immune signaling. Or even more recently, and this is a study that we have been involved, which was published this summer, where parasitic plants will be recognized by this innate immune mechanism. And I'll briefly come back to that later on. The other thing that I want to use this slide to illustrate, and this is why some of the molecules are highlighted in green here, is that there is a certain degree of recognition specificity, which means that not all plants are able to recognize all of the molecules I have listed here. And here in green, for example, I've highlighted the molecules which are recognized by the model plant Arabidopsis thaliana. And you can see that, therefore, this model plant is a very useful system to actually understand the diversity of this immune recognition and to understand downstream immune signaling, because it has the ability to recognize different non-cell determinants from a number of different macros. And we can use actually this specificity as well to study the evolution of the immune receptor, and as I will illustrate at the end of my presentation, to actually, for biotechnological purposes, to transfer some of this immune recognition specificity from one family of plant to another, and to increase thereby their disease resistance. So this is an extremely active field of research, which has boomed in the last 10 to 15 years. And this is a review, a schematic from a review we only wrote 4 years ago. And what you can see is that back then, we only know about half a dozen of such innate immune receptor from plant. But based on this knowledge, we could actually already conclude a few things. Genetically, we could show that these receptors are important for the overall resistance of the plant. And not only they are important during a compatible infection, which means when an adapted pathogen is actually infecting a given plant, but they are also contributing to what is called non-host resistance. And non-host resistance is a phenomenon which exists in plants, thereby a pathogen which is adapted to a given plant species very often is not able to infect another plant species. And this is something you can observe in your garden if you are growing vegetables, where you may have a tomato plant which is infected with a fungus. And very often the plant nearby, that could be a potato, for example, will not necessarily be infected. And this is because potato is actually containing, in this case, immune receptor that are making this pathogen not able to grow on potato. But these immune receptors are either absent for the tomato plant, or the pathogen has evolved to actually suppress this immune receptor in tomato. So we know now that these different immune receptors contribute to disease resistance, and they also provide broad spectrum disease resistance. Because of the conserved nature of the molecule which are recognized by this receptor, the type of resistance will be then mediated against a whole class of microbes. If you recognize, for example, chitin, which is one of the main constituents of the cell wall of fungi, you will then mediate resistance against mostly all fungi. But back then, as I pointed here, we only knew a few of these immune receptors. The other thing that we could conclude from this sparse knowledge is that biochemically, the immune receptor belong to two different classes of protein. They are either what we call a receptor kinase, which means it's a protein with an ectodomain here, involved in ligand binding, a transmembrane domain that will anchor this protein within the plasma membrane, and an intracellular kinase domain. This is a receptor kinase. The other class of immune receptor are what we call receptor-like protein. They basically look like receptor kinases, but they are lacking an intracellular kinase domain, or actually any signaling domain. And it has been for long hypothesized that in order to function, in order to trigger intracellular signaling, they will need to form a complex with a receptor kinase. And this has been shown now in a few cases. It was first illustrated here in the case of chitin perception in rice, where then there will be a ligand-induced complex formation between a receptor-like protein and a receptor kinase. So these are the two classes, biochemical classes of protein involved in immune reception, in perception in plants. So from 2012 to 2014, the number of immune receptors, of these PRs in plant, have almost doubled or tripled. And actually, this slide is completely outdated already. And we are currently writing a new review about the status of 2016. And this number has, again, almost doubled or tripled. And one thing which is important to realize as well is that now scientists have also been able to directly identify some of these immune receptors in important crops, such as wheat and rice, and been able to show that for many years, these immune receptors have actually contributed through classical breeding to the broad-spectrum disease resistance of this crop to important pathogens. So if now that we have a better knowledge about how these immune receptors are looking like at the molecular level or even within the genomic region where they may have certain signatures of evolution, we can go back to plant genomes. And what we realize is that plant genome have actually the potential to encode hundreds of these different immune receptors. And this is a major difference with our own immune system, of innate immune system, where we only rely on maybe 15, 20 such innate immune receptor, whereas plant most likely is relying on hundreds of them, each of them conferring a certain recognition specificity, enabling the plant to defend itself against all the many potential pathogens which are present in their environment. So in my lab, we basically try to address three major questions, or we have three major interests, if you want. We are interested in receptors, we are interested in signaling pathways, and we are interested in immunity. In terms of receptors, we try to identify new molecules which are recognized by the immune system, which would activate defense responses, and to identify the corresponding immune receptor. This is actually mostly linked to our interest in biotechnology, and I'll come back here, and how we can engineer broad-spectrum disease resistance. Most of our group is actually studying signaling pathways. Immune signaling, this is our prime interest, but also signaling involving growth and development, because we see a lot of similarities, or actually competition, if you want, between the two pathways. We do a lot of biochemistry to try to identify how this immune receptor within the plasma membrane are actually not acting as isolation, but acting as very large macromolecular complexes. And within these complexes, there is a lot of post-translational modification, and this is something that we are actively studying. What is the role of phosphorylation, for example, in the activation of this protein, and what is the role on the other end of ubiquitination to potentially downregulate this regulation? And I'll briefly illustrate this later. We also try to understand how, once this immune receptor complex is activated at the plasma membrane, they actually connect and activate downstream immune signaling component, and what is the identity of this component. At the moment, there is a black box, if you want here, how to connect perception of a non-self-determinant to the actual activation of immunity downstream. But as I mentioned, the class of immune receptor we are working on are receptor kinases. And this is actually one of the largest protein family in plants. In Arabidopsis thaliana, there are 600 of them. In rice, this has massively expanded. There are more than 1,000 receptor kinases. We barely understand what is their function, what is their ligand. But actually, thanks to our work on immune signaling, we have really good tools to try to understand globally how this important family of plant protein is working at the molecular level. And more recently, because this receptor are working within the plasma membrane, we also became interested in understanding plasma membrane organization. How this receptor get to the plasma membrane in the first place? How do they organize themselves within the plasma membrane? And what we recently observed is, rather than being homogeneously distributed within the plasma membrane, they are present as part of macrodomain. And we try to understand how this organization within this macrodomain in the plasma membrane is actually underlying signaling specificity in plant between immune signaling, in this case, and growth signaling. And also, thanks to this work on the interplay between growth and immune signaling, also recently we observed that there is a very important role, regulatory function of endogenous peptide. And this is a booming area in plant science, where in recent years, when people have been steering and re-annotating a number of plant genomes, they have observed that in the same way that plant genome contain hundreds of different receptor kinases, they actually express hundreds of different peptides, and the function for most of them is completely unknown. So our prime interest obviously is immunity, but you see it's going in many different directions, where we try basically to understand how plants use receptor kinases to sense the environment. This can represent the environment as being a potential danger, but it also could represent their microenvironment, which would be mediated cell-to-cell communication, and in this case, not only in the context of immunity, but also in the context of growth and development. The mechanism of disease resistance here, I often get this question, at the end, what is affecting the pathogen? We basically don't know. This is still one of the big questions in plant biology. In many cases, and this is particularly true for bacteria, we don't know what is killing the bacteria. We don't know what is blocking their growth, and this is a very active field of research. So as I mentioned, because we are studying receptor kinase, and this is just to illustrate that our research is not, again, confined to immunity. We try to address this very simple question that basically anyone working on a receptor in any system in biology at some point has to address, is how to transduce the perception of an extracellular input, in this case by a receptor kinase, into an intracellular chain of event that would lead to an optimal intracellular output. So we use two receptors to actually try to address this fundamental question. And these two receptors are coming from Arabidopsis thaliana, immune receptor. They are classical leucine-rich repeat receptor kinases. One of them is called FLS2, and the other is called EFR. FLS2 is the immune receptor for bacterial flagellum, while EFR, which is related to FLS2, actually recognizes another protein from bacteria, which is called elongation factor TU, or EFTU. And we are fortunate that we actually know the minimal immunogenic epitope within this protein from bacteria, which is sufficient to mediate this recognition. In the case of flagellum, this is a 22 amino acid peptide called FLAG22. In the case of EFTU, this is an 18 amino acid peptide, which is called alpha-18. And we could show that this peptide directly binds to the ectodomain of both FLS2 or EFR, respectively. We also know that these two receptors are important for the resistance against bacteria and plant. This is illustrated here, where, for example, in the model plant Arabidopsis thaliana, if we knock out both FLS2 and EFR, so where these genes are not functional in this plant, then these plants are becoming hyper-susceptible to bacterial infection. In contrast, what we have shown in the case of EFR, which seems to be innovation of the Brassicaceae family, which contain Arabidopsis thaliana, but also cabbage or mustard plant, for example, we show that if we transfer this gene from a Brassicaceae to another family of plant, in this case a Solanaceae plant, such as tomato, then the expression of EFR, the presence of this new immune receptor, is sufficient to confer disease resistance against bacteria. So what this work is showing is that this immune receptor are both necessary as well as being sufficient to confer innate immunity against bacteria. So this very simple schematic is actually summarizing 10 years of work by many different groups around the world. And what we now know is that, as I mentioned before, this immune receptor are not working in isolation. And despite having all the domain, the biochemical domain, they will require to fulfill their function, the first thing that this immune receptor are doing is to recruit another receptor. This receptor is also involved in ligand binding, but it is acting as a co-receptor. So we have here two receptor kinases, one which is a ligand binding receptor, and the other which is a co-receptor. There are then a lot of activation loops between these two proteins within the cell, and this is where probably the function of the co-receptor is required. It is here to basically boost the signaling activity of the main ligand binding receptor. In response to this increased activity, which involves phosphorylation, this is why the P is highlighted here, there is the activation of another kinase. This is not a receptor kinase. It is not present within the plasma membrane. It is present inside the cell. It's a cytoplasmic kinase. The most notable one is called BIC1, but I don't want here to come down to a specific name. And then, once this cytoplasmic kinase is being activated, this will be the job of this kinase is to transmit immune signaling, and then which will lead to immune output. So a lot of our work is simply to try to understand how this complex is working, how it is regulating, how it is getting formed. So, if I summarize some of this work for the past 5 years, the first question when you work on a receptor is to try to understand how the ligand is being recognized. And we're fortunate to have an extremely fruitful collaboration with two groups in China, in Beijing, the lab of Gigi Shai in Tsinghua University and Jiamin Zhao at the Chinese Academy of Science as well in Beijing. And this group had managed to obtain a co-crystal of the ectodomain of FLS2 here, together with the co-receptor BAC1, together with the FLAG22 peptide. And thanks to this structural information, we got a lot of answer for the question which we are trying to address. The first one, obviously, is that we could define the exact binding site of the FLAG22 peptide onto the FLS2 ectodomain. If you think about the LRR receptor, it is just a stack of repeats of different amino acids. And one could imagine that a peptide, which is rather small, could actually just bind on one of its repeat like this. What this work showed is that actually the peptide has contact point on this LR region on 14 different of its repeat. And this partially explained why previously using other approaches, colleagues could not identify the exact binding site for FLAG22 and 2FLS2. But to us, the most interesting observation was the finding that the FLAG22 peptide, through its C-terminal part, directly as well bind to BAC1. And this allows us to conclude that indeed BAC1 is a co-receptor, it is involved in ligand binding. And what this work showed is that actually the ligand is acting as a molecular glue that is enabling this two-actor domain to become in close proximity. And what at the moment we hypothesize is that thanks to this close proximity outside of the cell, this enables the two kinase domain within the cell to get in close proximity and to enable transphosphorylation event between these different kinases, and thereby to activate downstream immune signaling. So once a receptor has perceived its ligand, the receptor complex, which now contains as well the co-receptor, as I mentioned before, and this downstream kinase, need to be activated. And there's a big role here for phosphorylation event. And so for many years, we have actually been trying to map this different phosphorylation event to identify which exact residue get phosphorylated in response to ligand perception. And a few years ago, we could identify a single residue, which is a tyrosine residue, which was phosphorylated in response to the ligand and was strictly required for the activation of this receptor. If we block the phosphorylation of a specific residue, the immune receptor is completely non-functional. In addition, in this work, we could show that pathogenic bacteria, in this case Pseudomonas, is actually secreting within plant cell a tyrosine phosphatase. This tyrosine phosphatase is directly binding to the kinase domain of this immune receptor, and by doing that is actually blocking the tyrosine phosphorylation of this immune receptor, thereby blocking the induction of immune signaling and therefore enabling the increased growth of this bacteria in the extracellular space around these plant cells. So at the moment, we are still continuing this work and try to identify additional residues within this kinase domain of both the receptor and the co-receptor, which are phosphorylated in response to ligand perception, and to understand what is the function of this phosphorylation. In the activation, potentially in the deactivation, this is something we are considering, and also in the case of the co-receptor, in signaling specificity. Because one thing I don't have time to present is that this co-receptor is involved in many different signaling processes in plant, in both immunity, growth, but as well as development. And we have a key interest to understand how BAC1 is actually, this co-receptor, is able to mediate signaling specificity. So once this receptor complex is activated, obviously one has to understand how you then activate downstream signaling and how you lead to an immune output. And one of the immune outputs which is conserved in both animal and plant is a rapid generation of reactive oxidative species. This is often referred as the oxidative burst. It is thought to help killing bacteria or fungi, or at least to tighten the cell wall that surrounds plant cell and thereby blocking the ingress of different pathogenic microbes. We know the enzyme which is responsible for this production of reactive oxidative species is the NADPH oxidase. Again, they are rather conserved between plant and animal. In plant, they are called RBOH. And what we showed a few years ago is that the receptor complex directly activate this enzyme. They are part of a constitutive complex, protein complex, between all of these kinases and this enzyme at the plasma membrane. And we could reveal that this cytoplasmic kinase, which I introduced before, upon activation, will then directly phosphorylate the enzyme at 3 different residues. And this is strictly required for the function of this enzyme. If we block this phosphorylation, then the enzyme is not active. And this work initially came as a surprise or basically was shocking some paradigm because it was known in the plant literature that the major regulation of this enzyme was mediated by calcium. Calcium can directly bind to this enzyme and thereby activating it. But in addition, calcium will induce other kinases, which are calcium-dependent protein kinases, and there will also be involved in the activation. And actually, our model is not challenging this previous model. It is just illustrating that in order for this calcium regulation to be functional, we need priming of this enzyme by directly by the receptor complex. And this is a model that we have proposed and that we are currently testing, and all the results indicate that this is the case. And this will most likely also contribute to signaling specificity, because many different inputs can actually lead to a generation of reactive oxygen species. So this will be a way to directly link the activation of the enzyme producing reactive oxygen species to a specific receptor complex in a specific location within the plant cell. And finally, this is something that as humans, we tend to be biased towards positive regulation, something that leads to an effect. But many different research projects have taught us in recent years that the negative regulation of immune signaling is probably as important or more important than the activation of these pathways. And this is similar to what has been also recently observed in animals. This is actually a schematic from a recent review illustrating this point in human and mice, where this fine regulation of immune signaling is actually required for the overall homeostasis of the organism. If you activate too much or too early immune signaling, you are likely to cause autoimmunity, which is actually the cause of major diseases that humans are facing, the overactivation of some of our innate immune receptors. And if you respond too late, then you will enable the pathogen to win and then to cause disease. So we really need to find this fine balance when you activate immune signaling. And basically, in plant, it is the same. We know that an overactivation of immunity, for example, by having deregulated immune receptor, we call dwarfism, and also necrosis, which also can be observed in human, at least in the case of necrosis. Whereas if you don't have enough activation of immunity in plant, you will enable disease, which is represented in response to bacterial infection by these chlorotic lesions in yellow. So we have actually identified a number of these proteins which are involved in this negative regulation in recent years. And surprisingly, it appears that every single component of the pathway we are studying is under negative control. And this is actually something that plants love to do, is to have a signaling pathway which is ready to go and is just blocked by a negative regulation. And the way you activate the pathway is by negatively regulating the negative regulation, which is one of the best ways actually to turn very rapidly and reversibly a signaling pathway. So not surprisingly, given the importance of phosphorylation in our pathway, we identify a number of protein phosphatases, whose job is actually to remove this phosphorylation event. And we identify protein phosphatases which keep all of these kinases within the receptor complex under control. One that negatively regulates the ligand binding receptor, one that negatively regulates the co-receptor, and another which is negatively regulating the cytoplasmic kinase. In response to ligand perception, very often these phosphatases will either be deactivated or they will dissociate from the receptor complex, enabling the full activation of downstream signaling. Some complexity came as well in this negative regulation, and beyond protein phosphatases, surprisingly, we identify other kinases that in this case will not activate the pathway, but negatively regulate the pathway. We still try to understand how this is working. And also more recently, we also identify endogenous peptide, as I mentioned before, that plays a role in the negative regulation of immune signaling in plants. So this part of my presentation was just to illustrate the complexity of immune signaling in plant. But now what I would like to do is switch gear and actually to present how we can use some of this knowledge of this immune receptor to engineer crops which are more resistant to disease resistance. And the field, as a whole, of plant-microbe interaction actually comes to the realization, and this is what is discussed in this review, which was published in Science a few years ago, that now we know enough of the concept, at least, and we start to understand enough of the signaling pathways involved, that we are in a good position to turn some of this knowledge into direct application. And this is probably the field which is the ripest in plant biology to directly see the potential effect in the field of some of the discoveries from fundamental research. So when I started my lab almost 10 years ago, we already initiated some of this project, and we wanted to test whether some of these pattern recognition receptors, these plasma membrane localized immune receptors, could indeed be used to confer disease resistance in crops. And for this, we did a proof of concept study, starting with EFR, one of the receptors we are working on. I just want to use this slide to remind you that this is a leucine-rich repeat receptor kinase from Arabidopsis thaliana, which recognizes this extremely conserved bacterial protein, which is called EFTU, which is involved in translation in bacteria. And as I mentioned before, EFR seems to be evolutionary innovation of the Brassicaceae family, which is one aspect we wanted to use. And as well, it recognized a protein from bacteria which seems to be conserved in all bacteria. So what we wanted to do is to test whether we could transfer EFR from a Brassicaceae plant, in this case Arabidopsis thaliana, to a non-Brassicaceae plant. And in this case, we moved to a Solanaceae plant, either a tobacco plant or tomato. The first question was whether the protein will accumulate. It did. Whether it is functional, and we tested that by testing if it can recognize its ligand. It did. So then we wanted to see, will it confer disease resistance to bacteria normally infected these crops. And here we use a bacterium which infect wild-type tobacco. You see that there is a lot of wilting and yellowing in the leaves of the wild-type plant. And you can immediately notice that the plants that express EFR are much less affected by this bacterium. And this is also observed when we count the number of bacteria that we can recover from this plant. This resistance was not specific to this particular strain of bacterium, and we could use another strain, and we observe exactly the same result. We could also move to another genus of bacterium. In this case, we moved to Agrobacterium, which is causing these gold tumors, on stem of tobacco. In the plant that expressed EFR, we observed that there was no tumor, or when a tumor was forming, these tumors were much smaller. So this was already indicating that the type of resistance which was caused in this plant was of broad spectrum nature, at least when considering bacteria. We observe exactly the same in tomato. Here we had to use different bacterial pathogen. In one case, we use a Xanthomonas bacterium, and we also observed that the plant expressing EFR was more resistant to this Xanthomonas bacterium. And here using a very virulent bacterium, which is causing bacterial wilt, where you can see in tomato, where the tomato are completely dying, as if you've forgotten to water them, that the plant expressing EFR were much more resistant to this soil-borne pathogen, and also correlating to reduced bacterial growth. So as I mentioned, this was a proof of concept work. But based on this work, we could already conclude two things. The first one is that indeed the interfamily transfer of this pattern recognition receptor is possible. And this was indicating that all the signaling components downstream must be conserved between plant families, because the only thing we did here was to bring a new receptor. It's like plugging a new antenna, if you want, into a cell, and immediately connected biochemically with downstream components. But it also showed us that for biotech purposes, we can use PRs indeed as a mean to confer broad-spectrum disease resistance in crops. So this program has now evolved, and in a number of publicly funded collaboration around the world, we have tested the ability, in this case of EFR, indeed to confer increased resistance to bacteria in crops. The tomato project is the most advanced because this is the one we started with, and we have now field trials, and I'll just present that, in my next slide. We have also transferred EFR in potato. In both these cases, the main target is Ralstonia, this important soil-borne pathogen, for which no genetic or chemical control is actually currently existing, and it's a major pathogen in subtropical countries for this crop. We have also transferred EFR in apple, and currently we are transferring EFR in citruses. But one thing we wanted to test, now that we have shown that we can transfer receptor from one family of plant to another, is whether we could break the dicot-monocot barrier, so from one class of plant to another. And so for this purpose, we actually transfer EFR from Arabidopsis to two different monocot plants, in this case rice and wheat. And in both cases, we could show that EFR is functional and was indeed able to confer increased disease resistance. We tested the reverse as well, where we took an immune receptor from rice and then transferred it into a dicot plant, Arabidopsis, as well as tobacco. And here again, this monocot receptor was fully functional. Also in this work, biochemically, we tested the ability of this receptor to form a complex with immune component normally involved in immunity in this species, and indeed they were able to recognize biochemically this component. And this despite millions of years of evolution between dicots and monocots. Again, illustrating that the pathway we are studying in the first part of our presentation is an extremely conserved and ancient signaling pathway. So as I mentioned, for some of this project, we are very close to the field. This is the case for tomato. This work is being done in Florida, the University of Florida. And our collaborators have transformed EFR into a commercial tomato line, which is being sold in the US, which is very susceptible to Ralstonia. This field was infected to Ralstonia. And as you can see here, is that the plant that only express EFR as an additional gene are much more resistant to Ralstonia. We have also a project in apple. In this case, this is targeted against a pathogen which is causing fire blight. This is caused by a bacterium called Erwinia amylovora. It's a quarantine pathogen in most European countries, as well in North America. If you have an apple tree which is infected by this bacterium in most European countries, the only solution, and actually by law, you have to destroy the tree and to burn down the tree. It's a big problem in the north of Italy, central part of Europe, Switzerland, France, and big problem in northeast of America and Canada. It's called fire blight because once infected, the plant looks as if it went through a fire. This apple plant is a wild-type apple plant. The infection was performed here, and you see that within a few days, the rest of the plant completely died and burned. The plant here that express EFR have been infected. The infection was performed here, and you see that the plant completely survived. And we hope very soon that we can perform field trials as well with these trees and to see if this is maintained in the field. So although we're the first to test the ability of transferring this immune receptor from one family of plant, there are a number of different examples in the literature, and this is here some of them, and this is the one I mentioned here before, where a few months ago, colleagues in Germany could even identify an immune receptor, which is recognizing parasitic plant, and they could show that by simply transferring this immune receptor from one plant species to another, this was sufficient to confer resistance to parasitic plant, which is another major problem to agriculture. So I will just finish with a couple of slides where basically now we know that yes, we can transfer these PRRs from one family or the other. One dream, and this may take 10, 20 years or more, is actually at one point to be able to design immune receptor, to be able to have artificial immune receptor where we can predict we would like an immune receptor to recognize this particular protein, which is extremely conserved in fungus, for example, and be able to exactly design this immune receptor to recognize this epitope. This is a far dream, but we are working towards that. And at the moment, we are using EFR again just to try to force an immune receptor that we know to recognize its ligand, which has evade recognition. And as I mentioned, at the moment it doesn't work, but it's something we are working on. In addition, if we are serious about deploying this technology to improve disease resistance in crop, we need to know more family-specific PRRs and what are the elicitors which are recognized. And we have been involved in a number of projects in recent years to actually identify a number of these new receptors and the molecules that they recognize. And in order to make this study to identify immune receptor in crops where tools don't necessarily exist, or to assist breeders to see if the resistance we observe in the field actually correspond to what we are studying in the lab and what we call PAM-trigger immunity, we have to develop bioassays, toolbox that people can use. We like this bioassay to be cheap and high throughput. And again, we have been involved in a number of research studies to actually develop such bioassays. And we can now measure them in crops as diverse as citrus, grape, wine, barley, and brassica. And this list just expands with our collaborations. So in order to identify new PRs, people typically use genetics. It is still the best choice, but it is restricted to plant species where these tools are existing, which at the moment is working extremely well in Arabidopsis thaliana, in tomato, and maybe rice. So in recent years, we have also been developing a number of biochemical ways to go to identify this receptor. And this is based on our knowledge gained through our fundamental research to understand immune signaling, where, for example, by revealing the importance of the co-receptors and the fact that we'll only form a stable complex when the specific ligand is present, where we can use these co-receptors as molecular bait to go fishing, if you want, for the receptor that will form this ligand-induced stable complex. Equally, if we know a ligand which behaves nicely biochemically, mostly meaning a protein or a peptide that we can label, we can use the ligand itself to go fishing for the corresponding receptor. And all of these approaches will then depend on proteomics to identify the receptor which has been enriched with this method. And also more recently, we have been developing high-throughput cell-based assay to actually screen a number of potential immune receptors based on genomic information. And this is something under early development. And this is my final slide, and it's just a note about durability. And durability is a difficult word. It's something that often people use in a qualitative way by saying this is a durable or non-durable resistant. And I think durability is more quantitative word. You are more or less durable. And sadly, the pathogen will always win. They can evolve much faster than the plant can do. And so it's our work as biotechnologists to actually move as fast as possible to keep up with the evolution of the pathogen. So there is no silver bullet for durability, and there is no way to predict how much any strategy will be durable. It can be that a new pesticide put on the market by Syngenta within a year, to name one company, within a year will fail. It is the same for breeders deploying a new resistant gene in the field. It can fail within 2 or 3 years. This has been shown over and over the years. But there are different approaches that, as a community, people are discussing that could improve the chance of durability. There is a possibility to combine different type of immune receptors with different mode of actions, combining not only this immune receptor at the surface, which I introduced in my presentation, but together with this intracellular immune receptor within the cell. And this is something we are currently testing again in the US, where we're trying to combine a number of different immune receptors in tomato and to see if they confer additive resistance and whether in the long term they will be more durable. This could be combined with genome editing, which offers now the chance to actually mutate with very small disturbance to the host genome, genes which are required for the infection, which may be involved in nutrient acquisition, for example, to the pathogen. There is still a place, obviously, for classical control involving either biocontrol agent, and there's a lot of research and interest into that with the boom of microbiome, which is equally the case in plants, and where a lot of companies or labs are trying to identify microbes from the soil that will help when applied to crop to make the plant more resistant to pathogen. And obviously, I don't think we can get rid of chemistry. We cannot get rid of pesticide or fungicide. But we can massively reduce the spray and their usage. And most likely, using all of that together will make agriculture more sustainable, and we'll have crops which have more durable resistance. So with that, I want to thank you for your attention, and I want to thank my lab, and many of them actually sitting here today. They're great people, and it's a joy every day to work with them. A lot of previous lab members, and I cannot list all of them here, and all of them have stayed in science, and many of them are now leading their own research group around the world. And a lot of our work, as you can imagine, is depending on the excellent support services we have at TSL, and we have very close interaction with the proteomics group at TSL, and also with bioinformatics and all the other support services at TSL. We have a number of different collaborations around the world. I don't have time to list all of them here, but we are an extremely collaborative laboratory, and this is exemplified in all our publications. And obviously, none of this work would be possible without funding. We always need money. A lot of our funding, generous funding, is coming from the Gatsby Charitable Foundation. And as David mentioned, we also have funding from a number of European agencies. So thank you very much.
Thank you very much, Cyril. It's a pleasure for me to now say a few words of thanks to you on behalf of the university. And for those who don't know me, I'm Phil Gilmartin. I'm the Dean of the Faculty of Science. So it's a great pleasure to be able to say a few words now. I think tonight, Cyril, you've taken us on quite a journey starting off by placing your work and the work you were going to talk about in the context of the human immune system and human immunity, which is something that everybody will be familiar with. And then you took us on a journey through plant immunity, which I think will have revealed many insights to people that will have been some surprises, as well as some real insight into the molecular mechanisms that you've managed to decipher through the work that you've done. But what I think was particularly exciting and thrilling to hear at the end was how you've taken that fundamental science and applied it to be able to test in crops, to test the ability of these different molecules to have an effect that is going to be of beneficial use and is addressing one of the real challenges of the world, and that's sustainable food security. So thank you very much. It's been a very, very exciting and invigorating talk. I think what we should do now is open the floor to any questions. I'm sure there'll be some questions. We have time for a few questions before the reception starts. So if we've got questions, maybe we could wait for the microphone to reach. And I think Colin here has the first question.
Thanks. Cyril, you showed that very nice crystal structure of the FLAG22 and the receptor and the co-receptor. But I was wondering, in the native flagellum protein, does FLG22 make an accessible epitope? And does the FLS2 receptor recognize the native flagellum protein or a breakdown product in the cell?
So it's a very good question. Actually, it's not for this reason, but I have a slide that will help me to illustrate that. So the flagellum is actually the main building block of the motility organ, the flagellum. And within this flagellum, the way the flagellum is organized, the epitope is not accessible for recognition. However, within the monomer, the epitope is available for recognition. And what has been shown in a few cases is that as soon as a bacterium is landing on the plant surface and is managed to penetrate within the plant tissue, it is actually shedding off its flagellum. And then the flagellum gets degraded. And so this would release then these monomers that are able to be recognized. So that's for the first part. The second part, yes, the full-length flagellin, so the full protein, will also induce FLS2-dependent immune responses. It is just that the epitope is sufficient.
And also, while I've got the microphone, with EFTU, if you could say something about that, because of course it's a cytoplasmic protein. It's locked up inside the bacterial cell. So again, how do those immune receptors get to see it?
Yeah. So for many years, this was obviously a question I was very often getting, and I used to say, look, there could be two mechanisms. Either EFTU is secreted in a non-conventional manner, and there are evidence in some proteomic studies that EFTU will be released in the extracellular medium, so that would be one way. The other way is actually, this is an extremely sensitive recognition system. EFTU will be recognized at the picomolar range. And so you always have bacteria will get lyse, especially it's quite a harsh environment, the apoplast of plants. And so this most likely would release enough EFTU to be recognized. Once I read that an E. coli cell, 90% of its protein is actually EFTU in terms of the mass. But recently, actually, a few months ago, there was a publication showing that EFTU is present in outer membrane vesicles produced by bacteria. And they have shown that the major eliciting protein within this vesicle is EFTU, and that this is now the most likely hypothesis that our EFTU become available for recognition.
Can you say a bit more about these endogenous peptides that you mentioned in passing there and what their role might be? Are they linked at all with homeostasis and perhaps preventing the autoimmunity?
Yes, so I don't have any slide on that, but that's exactly, so through a genetic screen, so in a completely unbiased way, we identify a protease, a subtilisin protease. And when we look for its substrate, which was a negative regulator of immunity, and then when we identified substrate, it turned out to be a propeptide, which was then cleaved. And the result of its cleavage is an active peptide, which once applied to the plant is inhibiting immune signaling. And we could also confirm that genetically. And we now think that this is part of a negative feedback loop, because it turned out that the processing of this propeptide into this active peptide is increased in response to PAM perception. So most likely, in response to PAM perception, you activate immune signaling. Somehow this enzyme becomes activated. This peptide gets processed. It binds to a receptor that we identify, which has nothing to do with this immune receptor. And then it's blocking immune signaling. So it's part of a negative feedback loop. You induce, you block.
So, Cyril, you talked about designing receptors or being able to make them recognize things they might not otherwise have recognized. Could you envisage a situation where you might even then be able to change the location of the signaling pathway? So with like pathogens like viruses or phytoplasmas that end up intracellularly and might evade the plasma membrane, do you think you could just move the whole signaling pathway eventually?
To move them intracellularly, I'm not sure, because the LRR will not necessarily fold correctly. but to activate using other receptor some of the pathway, possibly, but it would need to be fused to other type of immune receptors.
Could you say a little bit about the variability, the natural variability of some of these PAMPs, Flagellum, for instance? You mentioned that bacteria will evolve and always outcompete and always be cleverer than us. But is that the case?
Yes.
Isn't Flagellum really quite stable? And if this type of resistance you're talking about is really the big shakes that it is, why haven't we seen that already? Why don't we see the evasion?
So we've seen it, and it's actually still happening. So there is also evidence on field studies that this pathogen is still evolving. So all of these proteins, Flagellum or EFTU, are extremely conserved proteins within bacteria. So which means if you would take 1,000 bacterial genome from many different genera, EFTU would be mostly conserved. If now you start to look at bacterial pathogens which have evolved to infect plants, you will start to see a few species which have mutations in residues which are required for recognition. For EFTU, it's quite rare because EFTU is so important, so conserved that maybe this will lead to a lot of deleterious effect. For Flagellum, there are now quite a few examples of symbionts, for example, so rhizobium, which infect legumes, as evade recognition by FLS2. This is a case for Mycobacterium as well. It is a case for Ralstonia. But interestingly, and I can again use this slide by chance, is that what people have observed is that the whole Flagellum at sequence level, nucleotide sequence level, will still be conserved, but except in the epitope, which is recognized. And there you will have evidence of positive selection. So indeed, there is a pressure on the pathogen to evade recognition. And this is why none of what I've showed you today is a silver bullet. I'm not saying you can deploy one of these immune receptors and that's it, we have solved the disease problem. But we can now understand which molecules are better to be targeted. The EFTU, it's much more difficult for a bacterium to evade recognition. So maybe EFTU is a better one than FLS2. But we need to use many different strategies to impede, if you want, the chance of the pathogen to evade.
Is there a molecule you can say that if only we could target this one, that would be the silver bullet?
Well, it's easier. So, obviously proteins are encoded by genes, so it's easier if you want to evolve. Carbohydrates, you would need then to potentially first, a change in the enzyme that would need to then lead to a change in the structure, let's say, of LPS. So maybe it's better to target recognition of lipids or sugar. At some point, the pathogen will evade, but it may take longer.
Well, thank you all very, very much, and thank you, Cyril. I think we ought to draw the questions to a conclusion now, given the time. There is a reception outside. So at this point, we say goodbye to our live stream audience, those who joined us by the live stream, and for those of you who are here, thank you for coming. And I can invite you to the reception in the lobby. But finally, can we just thank Cyril once again for a superb inaugural lecture? Cyril, thank you very much indeed.